Hello, guys. This is the second part of the lecture on B cells and the B cell receptor. As you can see here, this is a B cell with B cell receptor. This is also called immunoglobulin. The B cells are the major cells in our body that produce immunoglobulins, but these immunoglobulins are also called antibodies. So the B cell receptor actually is an antibody which is expressed on the surface of the B cells. And as I told you in my previous lecture, that either this is IgM or IgD, and then this IgM and IgD, it would find its perfect partner in the form of some antigen, which can be of bacterial origin or a viral origin or some other antigen presented via dendritic cells or directly come in contact with the B cells. And that interaction actually is a trigger, for the pathways that would lead to changes in the B cells, different mechanisms that are in place in the B cells. And the trigger is this kind of interaction or encounter. Where is this encounter taking place? It is happening in the lymphatic nodes because the B cells and T cells are there. They're not everywhere in the body. These are small lymphocytes, and these are taken from the blood to, these sites like the lymphatics where they would be trained. So the lymphatic tissue or lymphatics are the places where the B cells and the T cells are found, and these lymphoid tissues or lymphatic nodes are the training grounds of the B cells and T cells, where they are trained in how to interact with different types of antigens. So every B cell has one type of B cell receptor. Every T cell has one type of T cell receptor. And millions of different types of these B and T cells are produced in the body, but only one B cell or T cell would specifically bind to one specific type of an antigen. And when they find that specific antigen, then these B cells and T cells, of course, both of them, the principle applies to both of them, they will be clonally expanded. Just like you see, the B cell has turned into an effector cell, a functional cell that would produce the same B cell receptor which was expressed on its surface and which encountered, this particular bacterium. The same is being produced here or secreted here. This is secreted form of the immunoglobulin expressed on the surface of the B cell, and this secreted form of immunoglobulin is called an antibody. The difference between the two is that the B cell receptor has an intermembrane, anchor, which is produced when the B cell is going through this process of maturation of, in the lymphoid tissues, uh, in the bone marrow, and then it is anchored into the B cell membrane. So there is a transmembrane domain of this particular B cell receptor here, but the antibody, when it's produced, this antibody does not have that membrane, that part of the transmembrane domain which would anchor this antibody or keep this antibody, attached to the B cell. So this actually happens due to some alternative splicing of the genes that are responsible for coding these antibodies that we would discuss later. And first, let's discuss about the structure briefly. The antibody is a molecule of about 150 kilodalton. And if you see here, this part of the antibody, which is called the FC fragment or the fragment crystallizable. This is the crystallizable part of the antibody. That's why it's called the FC region or the fragment crystallizable. And this part of the antibody, the lower part that you see here, this upright part here, this FC portion also, you would have seen these FC receptors on different types of cells. So many different types of your body cells, they have receptors for recognizing an antibody. It means the antibody is going to attach to this cell, those cells with the help of this region. In the upper part of the antibody, if you look here, this part at the N terminus, this every peptide has an N terminus or amino terminus and the carboxy terminus. The carboxy terminus is here towards the Fc part of the antibody, and the N terminus is here towards the variable part of the antibody. So this variable part is for binding of the antigens. In yellow color, you see these are the light chains, and in green color, you see these are called the heavy chains. So these heavy chains is about a molecule of about 50 kilodalton, while these are about 25 kilodalton each. I'm talking about the light chains. So when you combine all these, it makes a molecule at about 150 kilodaltons. And these heavy chains and light chains, all of them are joined together with the help of disulfide linkages. So here is the antigen binding site shown in red. Both the variable region is found both in the case of heavy chain and light chains. So this red part actually is showing the variable region or the region which would be used for binding with an antigen. So this region is also called Fab region. This is the Fab region, and this is the FC region, the fragment crystallizable region. And above here, this is, the antigen binding region. That's why it is called Fab, or the fragment that binds to, the antigen. In the next picture, you see that an antibody can take different conformations in order to bind different types of antigen. For instance, this is a bacterium, and different types of epitopes are found on the surface of bacterium. These can be different types of peptidoglycans, etc., which are expressed on the surface of these B cells, and they can take different shapes. So these can be lipoproteins, peptidoglycans, any other type of macromolecules that are found on the surface of these pathogens like bacteria. And the antibody can, mold itself, or it is flexible enough to mold itself according to the shape or the structure of the epitope which is found on the surface of the bacterium. As you see, these rounds can be, the antibody can make a round pocket, or it can make a triangular shape, or it can make a rectangular shape, and it can, uh, flexibly do that. It can bend, or it can, make it both the variable regions closer to each other, or it can just expand it. And that is possible because of this hinge region, which is part of different types of antibodies, but some types of antibodies, they do not have this type of hinge. These disulfide linkages actually are holding these parts of the antibodies together. It's a Y-shaped molecule. Okay, so this hinge here is important for this flexibility, and antibodies that have these hinges actually are more flexible, and they can bind to antigens with many different in many different conformations. And the binding of antigens to antibodies is based solely on non-covalent forces. This is not a binding of a complement with the pathogen on the surface of the pathogens, which makes a covalent linkage there. These are just electrostatic forces, hydrogen bonds, um, van der Waals forces, or some hydrophobic interactions that would hold antigens and antibodies together. So this is not a covalent or permanent linkage that is going to hold the antigen and antibody complex together. These are electrostatic forces, just like the forces that are holding water molecules together, for instance, in the form of a body of a liquid or body of water. Just like that, these forces are also similar kind of forces holding these antigen antibody complexes together. Now, if you look deeply into the structure of this antibody, this is the constant region, and the constant region uh, you see this is CH1, this is CH2, this is CH3. On both sides, you see the same configuration. So CH1 means the constant region of heavy chain 1, constant region of heavy chain 2, constant region of heavy chain 3, and you see all of them linked together up and down with the same disulfide linkages. Here you see these the CHO, which indicates carbohydrates. The carbohydrates are attached to these heavy chains. And that's why I told you in the first lecture that these are glycosylated proteins or glucose molecules are attached or sugar molecules are attached to these antibodies. So the constant region also holds these glucose molecules. They are also attached to the glucose molecule. So there are different isotypes, of the heavy chain and different isotypes of the light chain. This is the light chain. Cl indicates the constant region of the light chain, and VL indicates the variable region of the light. VH is the variable region of the heavy chain. On both the sides, it is the same configuration that you see here. And there is a hinge region indicated by here. So the light chain can be of two types. It can either be kappa or lambda. In one antibody, for instance, this chain that you see here, it can either be kappa chain or uh, a lambda chain. There are two types of these chains, and it depends upon the shape of the chain that how does it look like, which look like a lambda or a kappa. The heavy chains can take many different forms that you see here. It can be mu, it can be gamma, it can be alpha, it can be epsilon. So there are different types of the heavy chains. It means this uh, heavy chains, they can confer greater variability to the types of antibodies. So mainly, if you have one kappa or lambda or the light chain that you see here, you have one, two, three, four, and five different types of the heavy chains. And the antibodies isotypes that we talk about, IgM, IgG, IgD, IgE, all these have different types of these heavy chains that you see here, and that's why they are different from each other. There are other differences with reference to affinity of different types of antibodies, that which one is more suitable or better to bind with particular type of antigen, or it binds with the antigen with increasing strength if certain mutations would take place and increase the binding capacity of the antibody. And so that is a phenomena that is in place there in order to make the antibodies more, uh, you know, active to attach with the antigen that it has encountered previously, and that's how the antibody switching, class switching would take place. So if you see here in more detail, in the variable region that you see here, there are some hypervariable regions indicated by these red dots. In the constant chain, in the variable chain, there are some particular hypervariable regions. And these hypervariable regions are the red dots that you see here. This is the region where the epitopes or the antigens actually are going to bind. So there is a great deal of variations in this region. That's why it is called hypervariable region. So these three domains, these are also called the complementarity determinant regions, because they are going to bind with the antigens there, and that complementarity between the antigen, and this part of the antibody, is determined. If that is a perfect match for the binding with this particular region, then the antibody would perfectly match with it, and it would bind with that particular antigen. So this is also called the complementarity determining region, or CDR. So an antibody can have CDR1, CDR2, and CDR3, complementarity determining region 1, 2, and 3. And these can also be called hypervariable region 1, hypervariable region 2, and 3. So this CD actually is different from the CD molecules that usually we call T cells as CD3 positive or CD4 positive T cells, CD8 positive T cells. Uh, that is cluster of differentiation. That's a different thing. Here, CD means complementarity determining regions. CDR means complementarity determining regions. We are talking about antibodies here. So this you can see the disulfide linkages also here, and these number of amino acids of different domains has also been given here. So this is self-explanatory. I don't have to go into the detail of it. Here you see these are different isotypes or classes of the antibody. This is IgG, this is IgM, IgD, IgA, and IgE. These are five different classes of antibodies. So in the initial lecture, I told you that the B cell receptor, which is found on the surface of the B cells, can either be this one, IgD, or IgM. So this IgD and IgM uh, later on, if the person is infected initially, you know, and the response, adaptive immune response is invoked, the B cell would majorly produce IgM because this is the B cell receptor which is found there, or it can also produce IgD. So the IgM that is found here, it's slightly different from other types of antibodies. If you look here, it does not have this hinge region which is found in IgG and IgD and IgA. They are hinge regions. It is more like IgE, which also does not have this particular hinge region. If you look at the number of the constant, heavy chains or light heavy chains constant domains, you would find out that IgM and IgE again are different. How many domains are here? One, two, three, and four. Four constant chain heavy domains are here. And if you look in the case of other antibodies, you would see that one, two, and three. 1, 2, and 3. In the case of other antibodies, you would see that there are only three constant heavy chain domains there. So IgM and IgE are different in that respect that they do not have this hinge region. So what actually should happen in that case, because the B cell has initially got this antibody IgM, so during primary infection or during initial phase of the infection, which is called the acute phase of the infection, the B cell is going to produce IgM, and IgM is does not have this hinge region. It means that it does not have the flexibility to open its arm and receive the antigen and bind to the antigen in many different conformations or angles. So there is another mechanism. Some of these antibodies are uh, pentameric, some are dimeric, and some are singular. So this IgM is pentameric. It would produce a pentamer in such a way that the tails or this constant region would be on the inner side and the arms would be outside. So this pentamer would produce a circular, structure, and that circular structure would be capable to bind with many different types of antigens in the infected tissue. And later on, this IgM would be replaced by IgG, this molecules. In chronic infection, when the infection proceeds to, uh, you know, uh, when the infection is a month old, for instance, or a couple of weeks old, then the body would replace this IgM with IgG. So the same, body, the same B cell actually is producing IgG instead of IgM. It has done this genetic rearrangement in order to and introduce certain kind of mutations that can introduce the hinge region in this particular IgG here and in this antibody here. It has reduced the number of the constant region that these are not needed. And it has also uh, this increased affinity of the variable region of the IgG to bind with the antigen. So the IgG, the binding strength of IgG with the same antigen against which this IgM was secreted initially by the B cell is more as compared to IgG or other form IgD, etc. So that's why class switching takes place. If you hear, if you look here at the isotypes of these heavy chains here, for instance, in the case of IgG is gamma, in IgM it is mu, these four domains that you see here. Again, you see here that it is delta in the case of IgD, and it is alpha in the case of IgA, and IgE, this chain is called epsilon. And if you look here about other differences in these five different classes of antibodies, these are the sugar moieties attached to the heavy chains. So you can see that there are different numbers of sugars attached to the heavy chains. So the intensity of the glycosylation of the heavy chains in case of different types of antibodies is also different. This is secreted in monomeric form. The IgG is monomeric, IgM is pentameric, IgD is again is monomeric. The IgA, which can be monomeric or dimeric, and IgE is monomeric as well. Uh, this table actually shows you the relative abundance of these antibodies in normal tissues. But the most important thing that you should remember is that why the class switching actually takes place, and what are the differences in these antibodies, different types of the antibodies that you see here. You can figure out by just looking at the structure of these antibodies that what are the differences there and what these differences actually are going to how these differences actually are going to affect the affinity of the antibody to bind with antigens. Why is the B cell producing different types of antibodies? You know, these antibodies, whether this is IgG or IgM type or class or IgD or whatever, they are going to recognize the same type of antigen against which this particular IgM, which was the B cell receptor present on the surface of the B cell against which this particular antibody was secreted. So IgM and IgG or IgD, they do not differ from each other in terms of binding with that specific antigen. They are going to bind with the same type of antigen. This variable region is not going to incorporate changes that would lead to um, identification or binding with a, a different antigen. You know, it's also always going to be to bind with the same antigen, but it just a different isotype would have more binding affinity with the antigen, and that is achieved with the help of different mutations that takes place in the antibodies, the genes that are encoding the antibody. And uh, the plasma cell, one can ask you a question that what would be the antibody produced by the plasma cells? The antibodies produced by uh, the plasma cells in chronic infections, for instance, that most probably would be IgG instead of IgM, because that those interactions have led to changes in the genetic architecture of the B cell, and the B cell would now produce more potent and more suitable antibodies or immunoglobulins for binding with the antigens. So in the next lecture, I will just tell you about this genetic rearrangement, that how does it take place, and how it leads to you know this kind of class switching or this kind of antibodies production, a little bit about the genetic details of that process. Thank you very much.